Did you know that you share about 60% of your DNA with a banana? I know, it sounds totally completely made up, but it's actually entirely true. If we rewind the clock far enough, you, me, bananas, and literally every single plant, animal, and fungus on this planet, we all share the exact same great-great-great-grandparent. We can trace our entire existence all the way back 3.7 billion years to a single common ancestor. Scientists call it LUCA, the last universal common ancestor. But here's the catch. LUCA was already a living, breathing microbe.
So, that brings us to the ultimate mystery for this explainer. If LUCA was already alive, how did it actually come to life in the first place? We are talking about the origin of existence itself here. How could non-living matter, stuff like lifeless stones, dirt, and water, transform into a living, breathing, replicating organism? Was there some kind of magical spark? Or is life really just one massive, slow-burning chemical reaction?
Okay, to answer that, we're going to break this monumental 2 billion year process down into 5 quick parts. 1. The life force myth. 2. Defining a living thing. 3. Complexity and replication. 4. Boundaries and metabolism. And 5. The grand cellular merger. Let's just jump right in.
Part 1. The life force myth, busting animal electricity. Now, up until about 200 years ago, pretty much every scientist out there firmly believed that a magical life force lived inside all creatures, which completely separated living beings from, say, a non-living rock.
But the collapse of that whole life force idea actually happened super fast. Back in the 1780s, Antoine Lavoisier measured the heat and carbon dioxide coming from a living guinea pig, compared it to burning coal, and basically proved that life operates on everyday chemical processes. Then, in the 1790s, Luigi Galvani accidentally touched a dead frog's legs with a steel blade and a brass hook, making them twitch. He was absolutely convinced he'd found that magical life force and called it animal electricity. But yeah, Alessandro Volta quickly proved it was just a regular chemical reaction between metals, which, fun fact, actually led to the invention of the electric battery. And then finally, in 1828, Friedrich Wöhler did what everyone thought was literally impossible. He artificially created urea, an organic compound found in urine, using completely inorganic laboratory chemicals.
And this whole history is exactly where we get the term organic chemistry. Originally, scientists labeled things like fats and proteins organic because they were only found in living things, assuming a magical life force made them. But when Wöhler and later Marcellin Berthelot successfully synthesized those organic compounds from scratch in a lab using just carbon, hydrogen, oxygen, and nitrogen, well, it changed absolutely everything. It proved you don't need a magical spark to create the building blocks of life, you literally just need chemistry.
Which brings us to part 2, defining a living thing, the chemistry of existence. So, if we are really just chemistry without any magical spark, what actually defines something as being alive?
You see, the transition from a lifeless rock to a living organism isn't just an on and off switch. It's more of a gradual spectrum. And to cross that spectrum, we essentially need to check off 4 crucial criteria for life. First up, complexity. Living things are built from incredibly intricate chemical compounds. Second, metabolism. That's the ability to consume stuff from your environment and turn it into energy. Third, boundary. You need a physical barrier separating you from the outside world. Think of skin or a cell wall. And fourth, replication. The ability to perfectly copy yourself, exactly like DNA multiplying. All right, part 3, complexity and replication, space warehouses and clay. Let's tackle the first 2 items on our checklist right now, complexity and replication. Where exactly did these complex chemical building blocks come from, and how in the world did they start copying themselves?
Well, the answer might literally be falling right out of the sky. Enter the panspermia theory. This is the hypothesis that the essential building blocks of life didn't actually start here on Earth, but were delivered to a young planet by heavy bombardments of comets and meteors. And the crazy thing? We actually have the proof. By using spectroscopy to analyze light from distant galaxies, scientists have found that these exact chemical building blocks are already just floating out there in space. We found over 20 different amino acids on meteorites, and get this, in space clouds 25,000 light-years away, we've spotted the exact sugars needed to build RNA and DNA.
Scientist James Lovelock put this whole concept absolutely brilliantly. He said, "The universe is like a big warehouse with all the spare parts required for life available in it." Earth didn't need to invent all the ingredients for life from scratch, it just needed to mix the cosmic spare parts together.
But hey, spontaneous complexity can totally happen right here on Earth too.
Take the famous Miller-Urey experiment back in 1953. They wanted to test the whole primordial soup hypothesis, so they filled a glass container with basic inorganic gases, ammonia, methane, water vapor, and then shocked it with electricity to simulate lightning on early Earth. And guess what? Within just a few weeks, the basic building blocks of life, like amino acids, just started forming completely on their own. Okay, so we've got the complex parts, but how do they start replicating?
Let's look at how we achieve that second criterion, replication. According to the clay mineral hypothesis, it happened in 3 relatively simple steps. Step 1. The naturally charged surfaces of common clay actually bound primary organic molecules together. Step 2. This clay gave those molecules a steady platform to concentrate and link up into these massive chains, which over time evolved into RNA. And Step 3. This newly formed RNA actually became capable of storing genetic information and, here's the absolute key, self-replicating. This RNA world hypothesis perfectly explains how early molecules could multiply, evolve, and eventually build the superstable proteins and DNA that we know today.
Part 4, Boundaries and Metabolism, Bubbles and Deep Sea Vents. Okay, we're halfway there. We have replicating complexity. Now we just need to figure out how these fragile, replicating RNA chains got a protective shell and the actual energy to keep going.
To really grasp how boundaries formed, just think about washing your hands with soap. Soap works because its molecules are two-sided. They have a hydrophilic part that absolutely loves water and a hydrophobic part that totally repels it. So when soap hits the water, those molecules naturally curl up into little bubbles, trapping the dirt inside. Well, in the early oceans, simple fatty acids did the exact same thing, automatically curling up to form tiny, protective lipid bubbles.
So the crucial turning point is that when replicating RNA accidentally got trapped inside one of these naturally forming lipid bubbles, a brand new boundary was created. This formed what scientists call a protocell. For the very first time on Earth, there was a rudimentary physical barrier separating the inside from the outside environment. But here's the thing, these protocells needed fuel if they were going to keep replicating, which brings us to our final required criterion, metabolism. The hydrothermal vent theory suggests that life really got its start down at the very bottom of the deep ocean. We're talking extreme heat and mineral-rich water just spewing out of oceanic vents. To survive down there, our little protocells would trap passing nutrients inside their lipid bubbles, extract the energy from the intense heat and minerals to build more RNA, and then basically release the waste back out into the water. Boom! Just like that, we have a basic metabolism. Which leads us to part 5, the grand cellular merger. How we became complex. So, look at that. We've successfully checked off all 4 criteria: complexity, replication, boundary, and metabolism. We have basic, single-celled life. But how did we go from microscopic little ocean bubbles to highly complex plants, animals, and humans?
Well, we need to look at 2 distinct categories of biology to figure that out. First, you have prokaryotes. These are your simple, single-celled organisms, things like bacteria and archaea. Second, you have eukaryotes. These are the complex multicellular organisms, which includes plants, animals, and yeah, you and me. So how did biology make the jump from that first category to the second? It happened through literally the greatest evolutionary merger in the history of the planet.
Imagine this. About 2 billion years ago, a population of archaea and a population of bacteria were living super close together in this primordial ocean. They had a mutually beneficial relationship going. Over time, they got so incredibly close that a bacteria literally, physically entered the body of an archaea. But here's the twist. Instead of being digested or destroyed, the bacteria just survived inside. It kept its independence. These 2 completely separate single-celled organisms permanently merged into one supercell, which ultimately became the ancestor of literally all complex life on Earth.
I know, it sounds like a crazy sci-fi theory, right? Until you look at the actual forensic evidence discovered by American biologist Lynn Margulis back in the 1960s. Think about this. Every single complex cell in your body right now is powered by a little cellular engine called a mitochondrion. Well, Margulis studied the DNA of our mitochondria and discovered that it is an absolute perfect match for the DNA of a bacteria called typhus. The tiny engines powering your body this very second are quite literally ancient absorbed bacteria left over from a 2 billion-year-old merger.
And this merger? It was the ultimate evolutionary power-up. By absorbing those bacteria, these new eukaryote cells essentially gained their own internal power plants, giving them access to 200,000 times more energy than a standard bacterial genome. With that massive, unprecedented surplus of energy, genes could just go absolutely wild, rapidly developing and fueling the exact process of natural selection that created every piece of complex life on this planet.
And honestly, this brilliantly illustrates the biggest takeaway of this entire explainer. The transition from a lifeless chemical soup to breathing, incredibly complex organisms wasn't just an on-off switch. It was a beautifully slow, gradual chemical spectrum that took 2 billion years of trial, error, and absolutely perfect conditions to finally get right.
It required the exact right temperatures, lipid bubbles coming together perfectly, and deep ocean vents firing at just the right time. But the most staggering part of all of this, the raw materials that actually kick-started this entire chain reaction are not even unique to Earth. The very amino acids and sugars that built us are still out there right now, floating in the distant clouds of our galaxy. It really isn't wrong to say that we are, quite literally, made of stardust.
Which leaves us with a massive, somewhat haunting question to end on. If the universe truly is just a giant warehouse of spare parts, packed to the brim with all the complex chemistry needed to spark existence, where else out there is life currently brewing? Keep looking up and keep learning.